In this video, I'll talk through the 2025 Higher Human Biology paper and explain how I would approach each question. Starting off in the multiple choice, we're asked about DNA molecules. We have got our phosphate at the 5' end. We've got our sugar, and this time it would be deoxyribose sugar. And then we also have our bases. Right, we're asked about sugar. It's DNA. That means it is deoxyribose, not ribose, because that would be RNA. The bond is the bond that holds together the two strands. That's between the bases. Those are hydrogen bonds. Peptide bonds are found between amino acids and a protein. But your strong covalent bonds that hold together the different nucleotides together to make the DNA strand. A is the correct answer there. DNA polymerase will unwinds and unzips. And it also joins nucleotides to make the new DNA strand. Looking through our options, so A is to do with the mRNA transit, won't be that. Adds DNA to the forming strand, that looks good to me. RNA polymerase is involved in making the primary transcript, won't be for that one. And then joining the fragments, that is ligase, joins fragments. There we go. Next one, we are asked to identify the correct statement about this graph with lots of bits of information. Survival rate, new cases, 50 to 59. Okay, so we're in this bit here. Let's go through the numbers then. So for survival rate, and this is where we have to keep our wits about us to be able to go the right way. This is where you get the survival rate. So any grey sections need to go to the right. Any of the black sections need to go to the left. Every line is going to be 1. So 91, 2, 3, 4, 5, and 6. 96 for the new cases. And what I'm doing here is I am making sure I do it in the same order. So it's survival first. And then the other side. Now this is a little bit harder to see. I think it's 5 boxes or 5 lines up halfway between, which would make that 10. Both of them can be divided by 2. And then 96 over 2 does give you 40. And then that also gives you 5. So it's looking like A is my correct answer. But we should check in case we've made a little mistake. Difference in survival rate, 60 to 69. And 70 to 79. Survival rate is read off of the right-hand column. That's 10, not 20. They've put that answer because if you went the wrong side, you'd get 20. But it's incorrect. That's 10%. 40 to 49. New cases. 50 to 59 new cases. Well, new cases, that's coming out at 10. And we'll hopefully be able to see here, this is above 20. It's looking like 23, but certainly not 20, so it's not double, so it's more than double. Group with the highest percentage of new cases, which would be over here, 15 to 39, has the highest survival rate. Well, that's incorrect because the next group along 40 to 49 has that highest level. 8 is still looking good to me. Moving on, what type of RNA can be found in the cytoplasm? Okay, let's go through the job. So mRNA carries the copy of the DNA code from the nucleus to the ribosome. tRNA is carrying the specific amino acids from the cytoplasm to the ribosome for them to get joined together. The rRNA is part of the ribosome, and ribosomes are found in the cytoplasm. So I'm going to say it's that one. Now, I'm not the most confident one about that last one, but however, if you look at our options, we can't have mRNA and tRNA alone, and they are definitely found in the cytoplasm. So then that would be helping me make my mind up to go for D as the answer, because M and tRNA are both definitely there. And we'll go for that. So that must, I'm taking that as being then it's part of the ribosome. The ribosome is found in the cytoplasm, so you'd find it there. is what I'd be putting. Here we're talking about transcription. So we are taking the DNA letters, A, T, G, C, and they're getting turned into the mRNA. So we need DNA, because that's what gets copied into mRNA. It's turned into mRNA. Now, for the same reasons as mentioned above, the tRNA takes the amino acids to the ribosome. That's part of transcription. And the same with the rRNA, it's the ribosome where the joining of the amino acids happens. So again, not transcription, translation. And that would make my answer B for that one. Next one, we're at PCR. We're told that 1 cycle is 2 minutes. How long does it take to get 128 copies of one DNA section? So we are just going to keep doubling and doubling until the point we reach 128, 32, 64, and 128. We have 1 cycle, 2 cycles, 3, 4, 5, 6, 7 cycles. And if 1 cycle equals 2 minutes, then 7 cycles is going to equal 7 times 2, which is 14 minutes. Now, I have actually now reread the question. It is saying that it stays at each temperature for 2 minutes. There are 3, gets heated to around 95 degrees to break the hydrogen bonds and separate strands, down to 55 to allow primers to bind to target sequences, and 75 for DNA replication to take place. So actually, I have been initially conned, because it is actually 6 minutes. So then it's 7 sixes, which gives us 42 as the correct answer. So most commonly, they'll tell you how long 1 cycle, that's the 3 stages together. They've been a bit sneaky here, but just about got away with it. Pharmacogenetics is to do with drugs and medicines. Personalized medicines. So I'm looking for something that says that. Okay, well, A, they're looking at how likely diseases are to be passed on. That is genetic screening. Next one is to do with using computers. That is bioinformatics. You might have had like IT class in S1, S2, where you're using the computers to do various different things, but informatics, IT, computer, that's what helps me remember that one. Drugs, dosages, that's medicines. Personalized medicine is the one we're wanting for it. Oh, and you're using computers again for the bottom one, so that will be, again, bioinformatics. Moving on. We want an increased production of isoleucine would be caused by... Okay, let's have a look. So if isoleucine was acting as a competitive inhibitor of number 2, well, that would stop this from happening, which would mean we can't get any of them. So we wouldn't get more of them. That doesn't sound right. Next one, negative feedback inhibition of theronine by isoleucine. Well, again, did that not then stop that from... Well, theronine's the part that's in it. It's not actually the enzyme that would be inhibited. So is that maybe just not a thing that can happen? Let's say no to that one. Right, if I had an increased concentration of isoleucine, well, that's not to do with the production of it. Yeah, you'd have more of it, but it's not making more of it. If you had more theronine, then more of that can get into the system and be converted through them. And we'll get to that last one as our answer for that. So I think they're trying to throw us off by talking about chemicals that are substrates instead of the enzymes. Perhaps here, vigorous exercise, not enough oxygen to cells. So we are into the fermentation process. And in humans, that's going to give you lactate. Acetyl group is what's formed in aerobic. The job of NAD is to carry hydrogen forward into the later stages. But in fermentation, you only have the glycolysis section happening. That means you wouldn't have hydrogen having been removed and then moved on to the future stages. So that feels like it's that one. It's just that simplistic early stages of them is what I would go for there. Calculate how many more times can fast twitch contract compared to slow twitched. Well, they haven't given us how many contractions in a minute. So maybe we need to do that first. Let's do that first. Right, slow says. So 1,000 milliseconds is 1 second. So this is 0.1 seconds. And then this will be 0.075 seconds. So one contraction, 0.1 seconds. And we need to get up to 60 seconds. I'm going to put 1 in the middle. A couple ways you can do it. The easiest way it works for any numbers, I'm going to divide by the first one to get it down to scale it to 1. And then I'm going to times by the second one to scale it up. We could have times by 10 and then by 60 or times by 600. I just find it easier to do that. Then whatever the number is on the left, we're going to do the same things. Divide by 0.1 times by 60. Is that 600? 1 is 0.075. Go through 1 up to 60. Divide by 0.075 times by 60. So 1 over 0.075 times 60. 800. Right. How many more times can it contract in a minute than the slow twitch? Well, it's 800 versus 600, which gives us 200, which will give us B as an answer. So a couple of things to watch out for. You need to watch out for the fact that they give us milliseconds, we want seconds, and also we have to scale up to 1 minute to compare them. Next one, we're talking about sprinters versus long distance. So about the sprinter. In sprinting, they don't have time to breathe normally. That means that they're going to be acting anaerobically, fermentation, without as much oxygen. Long distance runners have the time to breathe and get their breathing sorted throughout the race. That's going to be aerobic. So we need to focus on the sprinter. Myoglobin stores oxygen. They are then going to be lower because there's less aerobic respiration happening. So get rid of that and get rid of that. Energy sources are glycogen or fat. Glycogen is what the sprinters would be using. So that means that's going to be higher. Get rid of the lower ones, which we've already got rid of, to be fair. Mitochondria job is aerobic respiration. Well, if they're using fermentation pathway, then they would need less of them, making my answer A. And then if you were to focus on the long distance, it'd be the other way around. More myoglobin, less glycogen, more fat burning, and then more mitochondria. Moving on, we have got a graph, and we are asked to work out which of these is true. Okay, 1980, 1990. We are at that number and that number. So we must be going up in 2s for each box because that would be 10. So that would be 72 and then 86. It's gone down. It has gone down by 14. 14%. Sneaky, sneaky. It's gone down by 14 million per centimeter cubed, not 14%. Okay, then decreased by 60 million in the next two sections. So 70 right at the beginning, and then 2010, which is here, decreased by 60. Well, that's just not going to be correct because we're at 100. If you did decrease by 60, you'd ended up at 40, whereas we ended up above 40, so 44. So that cannot be correct. So that's gone down by 56, not 60. Current trends, 2025. Gonna see if this would work. So if we were to extend it, that's 5 more years after 2020. And then if we take this line and we extend it beyond there, and then I want to run this up to it touches, and then I want to run that across. Well, that's looking like it would be above that. That to me is looking like 24. So it's not going to be that one, have they maybe thought, I think what they've done there is if you took, so the gaps here, if you repeated that gap, you would be at 2030, and at 2030, it's looking like it's coming off at 16. I think that's maybe where that kind of false flag is came to try and throw us off a touch for there, but it's not going as far as that. You could have as well looked at how much it's gone down by in that time period. So in the last 5 years, so there we're at, let me get my eye in, so that's 30, 32, 34, 36, 37, down to 30. So it's dropped by 7. So the next 5 years, it would be expected to drop by another 7 to get 24, and then drop again, perhaps. But that's going to be the way for that one. Next one, for the average decrease, I would be wanting the decrease over the years. So it's just from the start to the end. So we've went from 100 down to 30, so it's dropped by 70. And this is where I always get myself muddled up when it's to do with these year things. You've got, how many years are we being inclusive of it? That'd be 50 years, and that would give us 1.4, which is the correct answer. That's going to happen as well. So a lot of kind of fiddling around the numbers to make them work, but that should be the correct answer there. Next one, got validity and reliability. Well, reliability is when you would repeat the experiment and you would get average results. Now, 3 groups of 4,000, quite big sample sizes, and they've repeated it. I'm going to say that's going to be reliable. In a valid test, you want to be doing only one change between the groups. I am reading here that they were given a different fertility drug. That should be the only difference. However, they've also, some of them, or many of them, have taken additional supplements. So that has not been controlling all the potential variables that would change the results. That's not going to be valid. That is going to be reliable results for that one. Ultrasound, so you have anomaly and dating. Dating happens earlier in the pregnancy, and anomaly is a little bit later. So 8 to 14 for dating, and then the anomaly scan is a little bit later at 18 to 20 weeks, giving the chance for the baby to develop a bit so they can see if there are any issues. That matches C here for this one. Right, next one. There are 3 outcomes. Now, I'm not sure exact letters to use here, but I'm going to go for just 2 different letters. So if we have healthy, if we have 2 of the healthy alleles, that person would be unaffected by the condition. If they have 1 healthy and 1 mutated one, they will have the trait, which affects them less than the full disease would, which would be done if they've got 2 of the sickle cell alleles. With those ones, when you've got 3 of them, that's described as being incomplete dominance. So instead of it just being someone has it or doesn't have it, they can have it, a mild version of it, or have the full-blown disease in this case, making that one there. Sickle cell is like a classic one for this with the trait. That's what I kind of jumped straight to that. You could look at things like, for dominant conditions, anyone who is affected, all their children would be affected. So if you look at here, we've got 2 people with the disease, yet they're able to have a mixture of they've got the trait, and they're able to have a mixture of unaffected disease and trait as well. If it was dominant, it would show all the way through. Sex-linked recessive would affect more males than females, but here we've got a bit of a split. In some questions, you'll need to actually think about, well, if it was sex-linked recessive, this would be an affected male, and then they would pass it on to that gene onto their female children, and it depends on the mom. But for this one, there are 3 outcomes. It's going to be the incomplete dominant one. Here we're talking about blood pressure and lymphatic systems. Explain the difference in blood pressure between points X and Y. So we're going from an artery to a vein. So arteries will be pushing the blood more quickly. So blood pressure is higher at X or lower at Y. So I can rule out these two because that's just factually incorrect. We're talking about the blood. So the plasma, blood has plasma in it. That's the part that gets squeezed through. And then any excess fluid gets reabsorbed by the lymphatic system. And that's looking like B for that one. Because that's saying if D was correct, you'd be having things going in this way, but no, it's squeezing, pressure filtration, squeezing the fluid through. Yeah, the capillaries as it's going through. That one. What have we got next? Next, not type 2, or may not occur with type 2 diabetes. This is the one that is typically diagnosed in adulthood, and they are less sensitive to insulin. Okay, so blood glucose would increase, high sugar detected by the pancreas. They can still produce it. Now, this is the problem part. So they have insulin, but their body doesn't respond to it in the way it's supposed to do. That's going to make that one the correct answer. If it was type 1, that would be the production part. So this one not for type 1. That's the one that's treated with insulin injections. 18, fine motor. Well, we have one signal coming in and it's splitting apart. That is divergent neural pathways. Summation of weak stimuli would be if those ones were all coming together, which is not what's happening. That's also converging, so they two mean the same thing. And then for reverberating, I would need to see signals going back and keeping that loop going, which isn't what's happening. So B for that one. Another numbery problem-solving question. Total number of individuals diagnosed with dementia increased every year. Let me just ping in the totals for us. I mean, if my maths is right, that's not happened, so I'm pretty sure that's wrong. I'm going to just do the rest of them just in case it comes up. I'm going to use those numbers for something else. Right, that one then is incorrect. More were diagnosed in 2016 than any other year. Highest value, nope, that's 2019. Just about did them all. More individuals, 51 to 80. So that's the combination of these two now. Brilliant. So they're saying that it was 2019 to 2016. Okay, so 2019, add those two together, you will get 10,723 versus 9631. That's looking correct. That is bigger than that. More individuals were diagnosed in 51, so in the first one, than any other categories. Well, they can't both be right. So then we need to check this set and the next set. So 19,377, and the next one's 20,957. So that is more in 66 to 80. So as soon as you can disprove any one section being higher than the first one, then you're all good to cancel it out, to ignore it. You're all good to say that is incorrect. And that gives us C confirmed. Next one, we're asked about predicting levels of drugs in a system. What's happening? So every minute, so here we're losing 2, we're losing 4, we're losing 6, we're losing 8. So if it consistently was broken down, we would expect minus 10, which would give you 6, which would give you an answer of A. So look for the pattern, follow the pattern, get your answer. Sometimes questions might have the exact same number every time, or there might be the gap is getting smaller, so you just want to fit your pattern as best you possibly can. Mast cells produce histamine as part of the immune response. That's just what I was asking for that. What are the other ones? So this is the phagocytes. Engulf and digest. You have got your B for bodies, B lymphocyte. During the inflammatory response, histamine's job is to dilate the blood vessels to get more blood so you can get more materials to help fight off any kind of potential infection that's happening. So it's the opposite way around. Here we're talking about white blood cells and we have to follow the patterns. Okay, initially within normal limits. So I want to go to the beginnings. So here we need to be between 5,000 and 10,000. Yes. No. I mean, it started there. So still in the running. And then it starts down there. So that one. This one. It's right on the edge. So I'm going to rule that one out. Yeah, I'm going to stick with that. Right, what did I do? 2 days later, it increased to 15,000. Well, that decreased, so not that. 15,000. I've already ruled out this one, but that bit's okay. And then that bit's fine. So B and D are the only ones still in the actual running. After 4 days, it had fallen to 3,000. So that has fallen, and that has fallen. And then it's gradually increasing within normal range. So we've made it back above 5,000. And actually here, this is now beyond that. That's beyond it. But then that is. So the only one that does everything correct is going to be D. So that one, so just take your time with that and that should be smooth enough. This one, we have 2 different axes. We have to be careful that anytime we're using the T lymphocytes, we are going up to the dashed line and over to the left. And then anytime we're using the full line, we need to go over to the right. Okay, 3 years, both the numbers. So let's go for it. 3 up to, let's do T first. So T is up to here. Run that across. That gives us 100 T lymphocyte. So not that, not that. And up and across. We would get 50. 8 is what I'm picking for that one. What they've probably done to try and throw us off is if we went the wrong way, we could quite comfortably think that that one was 10, the HIV, and then the other one would be 500. So answer B is if you go the wrong way and read the values off the wrong axes, which is not what we're going to do. Next one, ratio. White blood count. Oh, right. So what have we got? We've got 7,000 cells in 1 centimeter cubed. So that means we have 70,000 cells in 10 centimeter cubed. It's a 7:3 ratio. That is 10 parts in total. So we might actually be able to get away without doing any number crunching. So because it's 70,000, both these numbers, the 2 numbers have to add up to 70,000. Those don't, they only go to 7,000. That's there in case you don't catch, you need to scale it up by 10. So I'm going to rule those under instantly. It's a 7, bigger side is phagocytes. There must be more phagocytes than lymphocytes. So the only one that could work would be C. And that gets me my answer there. If it wasn't multiple choice, we had to do it. Well, 7 out of 10 of them are going to be your phagocytes. And then when you type that in to your calculator, we end up with 49,000, which is what I've already said. And the other part would be 3/10 times the 70,000. And that'll be, I'm not even going to type it in actually from the question. It's 21,000 for that. But if you just look at the sides of this one's higher ratio number, there's got to be more skewed to that side, which is where I got C from. Influenza, well, the flu is constantly mutating. It's changing its protein coat. That is, its antigens. Vaccines are something that help. Partial solution. These would fight off infection. And it's not that the memory cells in the body are changing. It's that the virus is changing. It's a mutated virus. So your body doesn't recognize it. And A would be the correct answer there. Moving on to section 2. First question is asking us about a somatic stem cell dividing. We are making identical copies of them. That makes that mitosis as our answer for the first one. The next one, they're asking us about production of gametes. The male sex cells are made in the seminiferous tubules of the testes, and the female sex cells, the eggs, in the ovaries. That would be either of the answers for that one. Here we're asked about differentiating into specialised cells. Well, the stem cells will switch on specific genes and produce proteins characteristic for that cell. So if it has to be a corneal cell, it has to switch on these certain ones. If some other cell, it would do a different set of proteins. It would make a different set of proteins. In this next question, we have to deal with the information in the graph. We're asked to describe the changes as we increase from 0 to 99 years. First section, from 0 to 17 years to 70 to 79 years, we can see that it's going to be increasing from 5 up to 700, the number of donors. And then above that, so up till 80, or so from the 70 to 79 range, the 80 to 99, we're going down to 394. So just find the section where it changes direction, stops increasing, now decreasing, and then deal with them as you go through. A benefit of using a patient's own stem cells, in every age bracket, there are more people having transplants than donating stem cells. So there are less donors than recipients at every age. And if you use your own stem cells, you do not have to wait for a donor to be found. You have your own, you can be your own donor. You can give the, you can provide the stem cells to help with the treatment. Research things to do with stem cells. Well, one would be to test out drugs. So investigating different diseases and seeing how they progress as well would be another use of those ones. Next one, we're asked about PCR, and we're asked about the base that is in position 3. That is across from G, which makes that that's going to make it cytosine. It's then asking us about why 2 primers are required and why do they have different base sequences. Well, each strand will have a different DNA code, different order of bases. So we need the 1 primer for each strand to do that. When it comes to temperatures, well, initially we go to the highest temperature in order to separate the strands. It then goes down to the lowest temperature to allow primers to bind. So I'm going to go for 55 as my answer there. And then it's heated up to around about 75 to allow the DNA polymerase to replicate the DNA. That's around the optimum for the heat-tolerant polymerase. In this next one, we're asked about the inheritance that's happening here. In this one, we're asked about the type of inheritance for haemophilia A. We can see it's on the X chromosome, not the Y. That means it's going to be sex-linked as opposed to autosomal. We're then told that 6,300 males have the condition, which is 75% of the total individuals. What I would probably do here then is, well, the females who have it will be the other 25%. And if we look there, well, we could scale from 75 down to 25 reasonably easily. You could divide it by 3, or you could divide by 75 times by 25. Either way, you're still going to get 2100. Another option could be, well, if the numbers are a bit more horrible, not as nice, we could work out what 100% would be. So if we divide by 75 and times by 100, the same on the other side, we get to 8,400. And then 25% of that, a quarter of that, is 2100, just the same. So 2 different ways, depends on what works better in your head. Next one, we are showing the order of the exons in this particular individual. We're asked about the chromosome mutation causing this change. There are 4 chromosome mutations we need to know about. That's translocation, inversion, duplication, and deletion. For translocation, we're looking for exons to have come from a different chromosome. So it'd be a different set of numbers. If you look at without and with, they've got the same numbers, but they're just not in the same order. So it's not going to be translocation. Inversion is when you take a section and you flip it round. That is what we're having. From 1 to 22 appears in reverse order. So that's going to be an answer there. If it was duplication, it would get longer with a doubling up of numbers already there. So you might see like 8, 9, and 10 twice in the section, which isn't happening. And deletion would make it smaller by removing some of the numbers, which is not what we've got in that one. When it comes to exons, exons are the coding regions of the mRNA transcript. Introns are non-coding, that's why they are removed. Then in the next one, why is the protein non-functional in people with the condition? Well, the exon order has changed, which means that the order of the amino acids will be different. And if you've got a different chain of amino acids, they won't fit together in the same way. The shape of the protein will be different, and it won't do the job it was supposed to do. When it comes to alternative RNA splicing, there are different combinations of exons joined together, which makes different mature transcripts, which means we get different proteins made at the ribosome. This little diagram here kind of shows that. The first one has all 5 of the exons. In the middle one, we're missing exon 3. And then in the last one, we're missing exon 4. So they'd go on and make slightly different proteins, or very different proteins. And this next one, we're onto the electron transport chain of respiration. Use the diagram to explain how the inner membrane is adapted to maximize ATP production. Well, you've got all the little folds in the quite a large surface area. That's going to increase the reaction and make everything happen more quickly. NADH is formed in a matrix, so that's the middle section. That's the citric acid cycle. Name another location in the cell where NADH is formed. Well, NADH is used at the end, used in the electron transport chain of the inner membrane, so it's not that. It's made in the first stage glycolysis, which happens in the cytoplasm. One roll of electrons. Well, we can see they are making their way through, along the membrane. They are high energy, so they provide the energy to allow hydrogen to get pumped across the membrane. And at the end, electrons, hydrogen ions, and oxygen combine to make water. So either of those ones should be enough. What does enzyme X do? Well, it's making ATP, synthesizing ATP, ATP synthase. Next question, we're asked about the effect of substrate concentration on the inhibition of catalase by an inhibitor, copper nitrate. First question, name a type of metabolic reaction. That would be either catabolic or anabolic. Catalase breaks down hydrogen peroxide, making it a catabolic reaction. So that just goes in there. Energy would be released in the process. Variables that are not already mentioned to be controlled. That would be things like temperature, which is not mentioned. So temperature of the solutions. pH of the solutions as well would be another thing. They tell us the concentration of the enzyme catalase and the inhibitor, copper nitrate, but they don't tell us the concentrations of those things. So the concentrations of them would have an impact on it. Other things could include the discs. The size of the discs could have an effect on how quickly they would move, or how big the beaker is. If you've got a very tall beaker, it would take longer for it to go down and back up. Would be another thing as well. Could be for those ones. When it comes to controls, they let you see what would happen if you didn't use the inhibitor. If you hadn't had catalase, we couldn't say what effect they have. So we can see that the copper nitrate is making it take longer in all but the last case. Without that, maybe that's just what it should have been. We wouldn't know for certain. So it allows you to compare without the inhibitor present. So it gives you like a baseline level you can do. Graph time. So your concentrations of hydrogen peroxide will go along the bottom. So typically if the graph and the table are made correctly, the left-hand side of the table will be the bottom of the graph. So concentration of hydrogen peroxide in percentage. We can then look at the numbers we've got. Well, there are values at 0.5, 1, 2, 3, and 5. So if we look at the first dot here, that's going to need to be 0.5. So we must be going up in 0.5s all the way across. At 0.5 with catalase alone, you're up at 120. Other axis is going to be the time it takes for the disc to float to the surface. If we go up in 20s, we can cover the full range that we need. To double check, 0.5 for catalase is 120. That's the same as it is on the table, which is good. Now we need to put in our numbers for the other one. So we're at 192. Every little box has to be 4. 4, 8, 12, 16, 20 works out for the bottom section. If you're ever unsure, you could do 20 divided by 5 because the gap is 20. There's 5 little boxes. Either way, we would end up with our points here. So that is at 92, 136, 72, 56, and then 28, which is what we're looking for. Then just join them up, dot to dot, gets us our graph for our marks there. A conclusion, well, as the substrate concentration is increasing, the inhibition is decreasing because we are speeding up the time it takes for the reaction to take place. Always answer your aim, concentration, substrate, inhibition. Mention both of them, that should pick up the mark. Next up, we're trying to predict what would happen if there was a 6% hydrogen peroxide. Well, we can see from with just catalase alone, above 3% does nothing. It stays at the same time. I would predict it would stay the same as well for this. So 28 seconds as our answer for that one. And this one, we're looking at how physical activity and body mass affects fertility. State the physical activity and body mass category and the resulting in the greatest chance of fertility problems. Greatest chance is 27. That's for low activity and obese. And next you have to work out the BMI. BMI is the mass over height squared. 77 over 1.7 squared. Calculator says 26.6. That's my answer there. If they have a moderate level and a BMI of 33, what's their chances? Well, it doesn't tell us the BMI value, but we should know that 33 makes the person obese. Above 25 would be overweight. So moderate obese, 18%, just like that. Next one, suitable treatments for a low sperm count. Well, one would be artificial insemination. That is when sperm samples are collected, combined, and then injected into the uterus. That will increase the chances of that sperm being successful in fertilizing. You can also use ICSI, intracytoplasmic sperm injection. That's when the head of the sperm is injected directly into the egg, and then those embryos would then be implanted into the female. So head of sperm injected into the egg. In this next question, we're asked about the human heart. We're first asked about the blood vessel X. We need to be able to recreate our pathway where the blood from the body comes back through the vena cava into the right atrium, right ventricle, out the pulmonary artery to the lungs, back in the pulmonary vein from the lungs to the left atrium, left ventricle, out to the aorta. That's the answer. Aorta. Here, next one. How can we tell that it's in systole? Well, I've circled here the two atria. Nope. I've done the AV valves. Nope. Here, I have circled the semilunar valves that are stopping blood from getting out to the pulmonary, stopping blood getting to the pulmonary artery or to the aorta. Atrial is the top chambers. Systole means they're contracting and we're pushing the blood into the ventricles. So the semilunar valves are closed, so blood can't flow out of the heart. And the AV valves between the atrium and ventricles are open, so blood could flow down from the atria into the ventricles. Talking about the impulses from the SAN node, that's the pacemaker. Well, that's going to cause, we're looking to cause the ventricles to contract. Well, the impulse is going to travel to the AV node, the node between the atrium and the ventricles. That sends impulses to each ventricle, and which then would cause each of the ventricles to contract. That should hopefully be enough for that one. Next up, can calculate the increase in heart rate during exercise. So let's find the heart rate before exercise. So I'm looking for a repeating section, which here looks like that. That gives me 4 boxes. If one box is 0.3, 4 of them are 1.2 seconds. I want to scale up from 1.2 seconds to 60 seconds. Divide by 1.2 times by 60. Do the same to the one beat, and you end up with 50 beats per minute. Other one, we've got a shorter time for one beat, which makes sense if you think that exercise you want more oxygen, more blood around the body, heart will beat more quickly, so shorter time. That is 2 boxes, so 0.6 seconds for one. Divide by 0.6, then times by 60, or just times by 100. That's 100 beats. It was 50, it's now 100. Difference is 50 beats per minute. The medulla is controlling the autonomic nervous system. And to lower heart rate, we want to follow the parasympathetic branch of the autonomic system. There's 2 chemicals involved. In the autonomic system, there's 2 chemicals involved. For parasympathetic, it's acetylcholine, and that's what's going to cause the lowering of the heart rate. If it was sympathetic for the heart rate going up, noradrenaline is the chemical released. In this next question, we're asked about how an arthroma develops within an artery. Arthroma are the buildup of fatty deposits under the lining of the lumen, which is the endothelium. So something like buildup of fatty materials under the endothelium. Cholesterol is one of those fatty materials that deposits, so you could say cholesterol instead. What's the stent doing? Well, the arthroma is squeezing and making the lumen narrower. The stent is pushing them apart. It's widening the lumen, which will increase the flow of blood through. That gives more space for the blood to flow through. So widen the lumen would be the answer for that one. And the next one we're asking about thrombosis and how it can cause a heart attack. We're not talking about making the clot. So making the clot is it ruptures the endothelium, clotting factors come, prothrombin to thrombin, fibrinogen to fibrin. Fibrin forms a mesh network that clots the wound. We're talking about the clot has moved. So a clot will be broken. A part of a clot or a clot will move, will be released, and it's going to block the supply of oxygen to the heart muscles. Coronary artery are the arteries that supply the heart muscle with oxygen. So it sounds a bit nice if I put in coronary artery for that one. Moving down, we're asked to use the graph to calculate the risk of an individual dying from coronary heart disease in Scotland. Well, it says, so looking at the graph, it's 125 per 100,000. The question wants 1 in something. So I need to take my 125 and scale down to 1. So if I divide both sides by 125, I end up with 800. And then that gives me a 1 in 800 as my answer for that one. Why do you do it as per 100,000 in a graph? Well, the countries will have different numbers of people. So you can do a valid comparison between them, taking into account the fact that there are different populations. So a valid comparison between countries with different populations. Next question is asking us for a use of cholesterol in the body. One use is it's used in the production of hormones and also as part of the cell membrane. Either of them should pick up the mark. Get rid of excess cholesterol. The high density lipoproteins take the excess cholesterol to the liver, which is able to process it and stops it from clogging up the arteries and other vessels. Next one, we're talking about competitive inhibitors. These bind to the active site of the enzyme and they prevent the substrate from binding to it. So that would, in this case, so the statin would block to the active site of the enzyme and the enzyme could not make cholesterol. That would bring your levels down, which is what they're wanting. Next one they're asking is about an autosomal dominant allele. Just to start off with any of these questions, I would write down all the different outcomes we can have. 2 dominant letters are going to be affected, 1 of each affected, and 2 little would be unaffected because it's autosomal dominant. If it was recessive, then it would be only the one with 2 little letters would be affected. And if it's sex-linked, we need to think about males and females separately. Right, what can we figure out from this then? Well, if we look at the case where 2 people who are unaffected have children, none of their children can be affected. 2 affected parents cannot have an affected child. If we then think that's when we're dealing with the homozygous recessive people. On the flip side, if we have 2 people who are dominant, 2 big letters, all of their children will be affected. Another outcome we could have is if we had 2 heterozygous individuals, they can have a mixture of dominant, they can have a mixture of affected and unaffected children. Because it's autosomal, not sex-linked, it will affect males and females equally. And there's probably some other stuff we can talk about here. We're quite open with what we've said. In a lot of questions, they'll narrow you in, they'll show you a family tree, and you need to go off of the information that's there. So because this person is affected and this one isn't, this can happen and that can happen as it goes through. In this next one, we're asked about the divisions of the nervous system. There's the central nervous system and the peripheral nervous system. Based on the space, the probably actually wanted us to put PNS instead of the full name. Y is coming off of the peripheral one. Peripheral has the somatic, so that's the one that controls the skeletal muscles, And then autonomic is the one that's controlling the heart rate and breathing rate. That's going into your parasympathetic and sympathetic branches. Next type of neurons, where there are motor, inter, and sensory. Sensory picks up information. Inter passes it on. Motor is connected to an effector, something that brings about a response. I see muscle. It is connected to our motor neuron, would be the answer there. Dealing with myelination, myelin is produced by the glial cells or glial cells. It helps speed up the speed of transmission of impulses. That's going to improve the coordination. If you're going to start throwing objects at children, the younger the child, the more likely they are to be hit by it. As their myelin increases, they will be able to dodge it a little bit more as they get older. Next one, we're talking about getting the information from the sensory organs into the short-term memory. That's encoding, where the information is converted into a format that your brain can store. To get things from short-term into long-term memory, we can do rehearsal, where we go over things again and again and again. You can organise the information using diagrams or techniques that help get everything neat and tidy. A message or a messy notes booklet is harder to keep in your head. We want to try and get this all sorted nice and neatly into your brain, giving us a better chance of remembering it for exams. You could also use elaboration, where you're looking for not just a set of words, but you're looking for connections between them, or maybe some more information that helps get it stuck in the brain. Storing memories, that happens in the cerebrum, or specifically the cerebral cortex of the cerebrum. That should pick up the mark for that one. In a serial list, this is showing that the earlier words and the later words are remembered better than the ones in the middle. That can be partly because you're paying more attention at the beginning, and then you realise that it's about to finish, and then you zone back in for this last couple of words. They're asking us to explain why did they remember more at the start and more at the end than in the middle. Well, at the start ones, you get more time to rehearse. So if you're going through your list and saying them again and again and again, there's more time for that to take effect in your brain. Short-term memory has a limited capacity. So as you're hearing more and more words, those ones will start getting displaced. You're still practicing the ones in the beginning, so those middle ones then start to get lost. They're asked about why it's unreliable. No, they're not. They're asking us how we can make it reliable. Well, we used a group of students. We'll use multiple groups of students, or repeat it with more lists of words. So check it out multiple times, and then you can look at average results and things like that as you go through. So here we're asked, what is one effect of activating the reward pathway? That's going to be the feeling of pleasure, that little hit of happiness that we get when we do certain activities. Dopamine, a neurotransmitter, can be removed either by enzyme breakdown or reuptake. Agonists mimic the natural chemicals, the natural response. So prolonged use of that means that the receptors are being stimulated more and more often. That can then lead to a desensitization. Random grouping, another protocol. It's drugs, so we can give placebo. So with placebo, that's going to reduce the bias. If people are going into a clinical trial and they know that they're going to get a drug or not get a drug, well, if you don't get given anything, you know you can't have had the drug. Whereas if you're given a placebo pill or the drug, you don't know which one you've got. Kind of leads us into the double blind idea, where both the researcher and participant don't know who's been given the drug or the placebo. So, it's not asking us what they are. Hang on. It's why they're important. So by nobody knowing which one's which, you can get more, you can get valid comparisons between them. Next one is asking about statistical significance. You need to have a large enough sample size, so it's not just down to chance. So like in adverts, when they're talking about they're bigging up their how performance of their new toothpaste or some sort of healthcare thing, they might often say like 75% of people saw a difference. If they're only using 50 people, then that's not quite as significant as having a larger and larger sample size. is what we're looking for there. Next up, we're talking about lymphocytes and infection. Cues found on the surface of lymphocytes bind to antigens. That's going to be receptors. They pick up on information. And if they get the correct one, they'll pick up they'll connect to the antigen coating of the pathogen, disease-causing microorganism. What's the evidence? Well, they're making antibodies, I presume. Yes, B for bodies, antibodies. Next, we're talking about phagocytes. That's like Pac-Man. They engulf and destroy. So they have little enzymes that they put out that then digest them and break them apart, stop them becoming a problem. One, memory cells. So they allow a faster response to a second infection compared to the first one. That's going to produce the antibodies quicker. And that could stop symptoms shown because it's already been dealt with before your body outwardly shows it. So it's a faster, more antibodies quicker than the first time round for that. Oh, T1's the killer T cells. They release proteins. So those proteins cause programmed cell death or apoptosis. I can't remember if that's part of the higher course or if that's from advanced higher biology. No, death. They kill the cells. They destroy the cells, as it kind of says out from that. So there's enzymes that come out and they cause the infected cells to die. Next one, we've got a 2-axis graph. We need to watch out for the fact that the number is the dashed line and it's read off of the left, and then the coverage is the other side and off of the full line. Vaccination coverage achieved when polio cases were 8,000. So 8,000. Polio, 8,000. So each box is 1. So 8 cases goes to this one. Now that happened in 1995, but we're asked for the vaccination coverage. So because it's the same year we're talking about, we can run up our line and then we can run it across to the other side. Comes out halfway between 80 and 70. That must be 75 as our answer. You can only go from the left-hand axis to the dashed line and then from the right-hand axis to the full line. Percentage decrease. Right, let's get our two numbers involved. So cases 1980. Cases is the full line. And it goes over to here. That's 53. And then 2010 is here. And again, make sure I'm going the right one. That's looking like a 2. So 53 to 2. And we want to do difference over original times 100. Take them away, 51. At the start of the period, it was 53. Times it by 100, turn it into percentage. And one decimal place, one round to 96.2. Use data to describe the changes in vaccination coverage. 1980, 2000. Vaccine coverage, so that's the full line. Okay, so it's increasing and then it's staying the same. So we're looking at that section and that section. Okay, so the turning point, the point where it changes is going to be 1990. Okay, so coverage is the full line off of the right. Let's run that along. So that is at 20. And then up here, it's that 75. So from 20 to 75%. And then from 1990 to 2000, it doesn't change. It has remained constant. That's the word I like to use with these ones. You could say it remained constant at 75, but I left them at 75. So if it remains constant, it stays wherever it was. So it remained constant would be my answer there. Next one, table. Oh, okay. So we need data in the table and graph. Simple whole number ratios. Global number to cases in this country in 2000. So one of the countries is there. So 2000. So the country was 90. So cases 2000 global. So 2000 cases is the dashed line. And then this way. So that is 3,000. Okay, divide by 10. Divide by 3. Must be that. So yeah, just find numbers you can divide them both by to get it simplified. Because we now have an odd number there, if you halved it, it would become a point, and it needs to be a whole number for a whole number ratio. Surprise, surprise. 85%. It's 2,000, right? So we need to have at least 85. So we are trying to work out 85% of 2 million. Now, percentage is out of 100, so I find it easiest to just do 85 over 100 and then times 2 million. We need to be careful with our zeros. So when I do that, we get 170, 0 and 3 zeros. So 1.7 million. And that would be that one there. If your calculator has a percentage button, which is quite easy to find, you could just type in what's that many percent, so press the percent button, and then times it by whatever the total was. So again, the 2 million, and that would give the same answer as well. I like doing it with over 100 because no matter what type of calculator, even if you're using like a phone calculator, it's quite simple. You should still be able to make that work. Why might it not be reached? So one of the things there in a developing country would be money. So poverty would be a barrier to stop that from happening. So people who are against using vaccines are the two ones that I would think of first. Now we get to the essay questions, and we're going to look at both the essay questions now. Okay, so we're going to write notes on the follicular and luteal phases of the menstrual cycle. Starting off in the follicular stage, FSH is released from the pituitary gland. Follicle stimulating, its job is to stimulate the development of the follicle. Next bit. The follicle then releases estrogen. Estrogen has two effects. It helps causes the endothelium to develop, the lining of the uterus. It also is going to make the cervical mucus thinner, which then makes it easier for the sperm to make its way towards the eggs. When that reaches a peak, it's going to cause a surge in LH. And then that surge in LH causes the egg to be released. So ovulation is going to happen as a result of that one. This is going to happen midway through. This is when it's going to go from follicular to luteal phase. The follicle continues developing, and it's going to develop into the corpus luteum. And the corpus luteum is going to release progesterone. Progesterone keeps the endometrium lining. It keeps it developed. It stops it from breaking down as it's going through. At this point, it depends on if the person got pregnant or not. Because we're talking about the menstrual cycle, this is presuming that the person has not fallen pregnant. A drop in the LH is going to lead to the corpus luteum breaking down. That's going to then produce less estrogen. And then because of the lack of progesterone, the endometrium lining is not maintained. It's going to start to break down. And then that's going to lead to the end of the cycle. Right, what I'm going to do now is I'm going to look in the marking scheme. I'm going to see if I have, hopefully, managed to get at least 9 of the points. Right, 1 mark for FSH from the pituitary gland, and it develops the follicle. So 1 mark for a combined first 2 sentences. The follicle is releasing, secreting they're saying, but that's releasing. So I'll take that. Stimulates the development, thickening, regeneration, proliferation's there as well. So yeah, that'll be fine. Any of those would work. We've got the outcome, do we? Affects the consistency. That's what I've said. Just the specific effect. High levels. Surge of LH, yes. Surge of LH triggering release of the egg. Ovulation. Ovulation is caused by the LH surge. That's in brackets, but yeah, I'll take it. Why not? Ovulation is partway through. It marks the start of the luteal phase, end of follicular phase. Midway point, giving it. I'll take it. Follicular, corpus luteum, progesterone turns into, develops, yes, gives out, secretes, yes, maintains the lining. So they've got the promotes the vascularization, so more blood vessels come together, or the other one I've said is the maintaining or thickening of it. So again, I'm all good with that. The negative feedback bit I've not talked about. So because there will be less FSH, so no more follicle, then that means no more LH, because what do I mean? Go back here. So the follicle's releasing estrogen. If you're not making a follicle, your estrogen is not going to go up, and then that's not going to cause the LH surge, and so on and so forth. I didn't really go there. Have got about the breakdown of the corpus luteum, and then because of that, there's less progesterone, and then that's going to cause the menstruation for that. So I've got enough points there to pick up my marks. There are a couple other bits I haven't put in, but the full marking scheme would show you that if you're interested. Next one, we're talking about the biology of controlling fertility and contraception. Okay, so we'll talk about the differences in males and female fertility. So females have cyclical fertility, and they're only fertile for a few days every cycle, every month. It can be identified by temperature and also the thickness of the cervical mucus. So it will be an increased temperature and also a thinner mucus. For males, males are continuously fertile and will continually produce sperm. Moving over to contraception, there are different methods. There are barrier methods of contraception, and these are going to prevent the sperm from reaching the egg. Some examples of these would be condoms and the IUDs. The IUD is also known as the coil. It's implanted into the woman's uterus and stops the implantation of embryos. Switching over to chemical methods, that would include the oral contraceptive pill, the mini pill, and the morning after pill. Both of them in turn. The first one has both estrogen and progesterone. Now, this estrogen and progesterone is synthetic, so made in the lab, and it's going to mimic the natural negative feedback loops. It's going to prevent the FSH and LH from being produced for that. And that's the kind of idea we need to get across with that one. Mini is progesterone only and is to do with the mucus. A mini pill contains progesterone only and causes thickening of the cervical mucus. And then the last one, the morning after pill, is the emergency contraceptive, and it prevents or delays ovulation and reduces the risk of the person falling pregnant. For that one, again, let's check the marking scheme and see where I've picked up some/all the marks. Okay, so we get 1 mark for females cyclical and males are continuous. So we'll put 1. It's kind of like a combination of them because it's in both of them. Oh, you then got a mark for about the differences in terms of always producing sperm and then only fertile a few months. So that's actually 1, 2, and 3. So like this part and this part for 1, this part for another, for that. Oh yeah, and you get the 4th one. So 4 within those ones, I've just kind of split it up. They've put the continuous and cyclical together, then fertile a few days, and then 1 for that. That looks so bad. Never mind. Physical ones, how do they work? Have I got 1? Prevent sperm from reaching the egg. Yes. 2 examples of them. Yes. Chemical methods. How many do we need? Any 2? I've got 3. Take the mark. What are they made up of? Progesterone and estrogen. Yes. Mimicking the negative feedback. Prevents the release. Yes. Progesterone only thickening. Is that just the 1? Yeah. Oh, so they're just calling it the progesterone only pill, but it's the thickening part that's picking up the mark. And then preventing the ovulation. They've also got potential mark for saying that it can be taken up to 72 to 120 hours after unprotected sex. It would be another bit, but I'm just going to leave it like that. That brings us to the end of the question and the end of the paper. Thanks for watching. Goodbye.